The function of the nucleus is to maintain the integrity of these genes and to control the activities of the cell by regulating gene expression. The nucleus is the control center of the cell. The nucleolus takes up around 25% of the volume of the nucleus. This structure is made up of proteins and ribonucleic acids. Its main function is to rewrite ribosomal RNA and combine it with proteins. The result is in the formation of incomplete ribosomes. The basic function of the cell membrane is to protect the cell from its surroundings. The energy made by the mitochondria is in the form of ATP. ATP is an energy currency that every cell in our body can use, and it keeps us alive. A major function of the Golgi apparatus is the modifying, sorting, and packaging of proteins. The main function of the smooth endoplasmic reticulum is to make cellular products like hormones and lipids. The rough endoplasmic reticulum is a series of connected flattened sacs that have many ribosomes on their outer surface. Ribosomes are found free in the cytoplasm or bound to the endoplasmic reticulum to form rough ER. The cytoskeleton determines the shape of the cell. Vacuoles are storage bubbles found in cells. They might store food or any variety of nutrients a cell might need to survive. The purpose of the lysosome is to break down or digest things. A flagella allows the cell to move around. The cell wall surrounds the cell membrane and provides the cells with structural support and protection. Chloroplasts are organelles found in plant cells and eukaryotic algae that conduct photosynthesis. Chloroplasts absorb sunlight and use it in conjunction with water and carbon dioxide gas to produce food for the plant. The cytoplasm is a jelly-like fluid that fills the cell. A centriole is a small set of microtubules arranged in a specific way. There are 9 groups of microtubules. When two centrioles are found next to each other, they are usually at right angles. The centrioles are found in pairs and move towards the poles. Poles are at opposite ends of the nucleus when it is time for cell division. A centriole divides the cell. There are 5 certain steps in the process of mitosis. There's the prophase, the metaphase, the anaphase, the telophase, and the interphase. In the prophase, the cell gets the idea that it is time to divide. First, it has to get everything ready. You need to duplicate the DNA, get certain pieces in the right position, and generally prepare the cell for the process of mitotic division. In the metaphase, all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the centrioles send out specialized tubules that connect to the DNA. In the anaphase, the separation begins. Half of the chromosomes are pulled to one side of the cell, half go the other way. In the telophase, the division is finishing up. This is the time when the cell membrane closes in and splits the cell into 2 pieces. You have 2 separate cells, each with half of the original DNA. The 5th phase, or the final phase, is the interphase. This is the normal state of the cell. You suppose that when it comes to cell division, you could call this the resting state. It's just going about its daily business of surviving and making sure it has all the nutrients and energy it needs. It's also getting ready for another division that will happen another day. It is duplicating its nucleic acids, so when it's time for prophase again, all the pieces are there. If an animal cell didn't produce centrioles, the chromosomes wouldn't be able to consistently sort correctly. The DNA would be divided unevenly, and the 2 daughter cells would not be identical.